Hi viewers, welcome back again to one more other lecture session from this channel. So in this lecture session we will be studying regarding the concept of lac operon from the molecular basis of inheritance in detail. So here the regulation of gene expression, it refers to a broad term which involves the expression of genes at various levels. Is it clear? So let me write it here. Regulation of gene expression. Regulation of gene expression refers to a broad term that involves the expression of genes at various levels. So here each and every gene, once when it undergoes expression during the transcription mechanism. So here once when the particular gene, once when it undergoes expression during the transcription mechanism, it synthesizes in single stranded mRNA. So let me write it here. It synthesizes in single-stranded mRNA. And here the specific mRNA which generally synthesizes during the transcription mechanism, once when it participates in case of the translation, now here it is capable to induce the synthesis of polypeptide chain or protein. So let me write it here. During the translation mechanism, the specific mRNA, once when it participates in case of the translation mechanism, here the polypeptide chain or protein is synthesized. Polypeptide chain or protein is synthesized. Is it clear? So here the regulation of gene expression in case of eukaryotes, it involves various strategies. So here what are the various strategies which involves the expression of genes in case of eukaryotes? Here the very first strategy is nothing but what? Formation of primary transcript. Is it clear? So let me write it here. Regulation of gene expression. Regulation of gene expression in eukaryotes. Involves various strategies. So here the very first strategy is nothing but what? Formation of primary transcript. So let me write it here. Formation of primary transcript. Right? So here what is primary transcript here? The specific eukaryotic mRNA which generally synthesizes during the transcription mechanism, here it is termed as what? Primary transcript. So here the specific primary transcript, it not only contains the coding sequences, and in addition even the specific eukaryotic mRNA which generally synthesizes during the transcription mechanism, it also contains the non-coding sequences known as introns. Is it clear? And here it is termed as what? Formation of primary transcript during the transcription mechanism. So let me write it here. Primary transcript, it contains both exons and introns. So here after the successful formation of the primary transcript during the transcription mechanism, now here the specific primary transcript, it undergoes specific processing mechanism. Is it clear? So let me write it here. RNA processing. So here the RNA processing mechanism, it involves the various strategies like splicing, capping, and the other one is tailing. So here what is splicing here? So here splicing, it is the mechanism or it is the process through which the non-coding sequences or introns which are a part of the eukaryotic mRNA, they get successfully spliced out. Is it clear? Splicing is the mechanism or it is the process through which the non-coding sequences or introns, they get successfully spliced out from the primary transcript in order to yield the secondary transcript or mature RNA or intron-free RNA. Is it clear? And here the other two strategies, they include capping and the other one is tailing. So here what is capping and what is tailing here? Capping and as well as tailing, they are required for the successful export of the eukaryotic mRNA from the nucleus to the cytoplasm. Is it clear? As we all know, in case of eukaryotes, the transcription mechanism, it occurs in nucleus, and whereas the translation mechanism, it occurs in the cytoplasm. So here after the successful synthesis of the eukaryotic mRNA in case of the nucleus, the eukaryotic mRNA, it has to export from the nucleus to the cytoplasm in order to participate in case of the translation. But here prior to the export of the eukaryotic mRNA from the nucleus to the cytoplasm, the specific eukaryotic mRNA, it has to undergo specific modifications like post-transcriptional modifications, which includes 5' capping and the other one is 3' tailing. Is it clear? So let me show it here. So here let us assume it as the nucleus here. Nucleus. And here it is nothing but cytoplasm. And here let us assume it as the eukaryotic mRNA. Eukaryotic mRNA. So here the specific eukaryotic mRNA, it has to export from the nucleus to the cytoplasm in order to participate in case of the translation. But here prior to the export of the eukaryotic mRNA from the nucleus to the cytoplasm, the specific eukaryotic mRNA, it undergoes 5' capping and as well as 3' tailing. So here what is 5' capping here? During the 5' capping mechanism, successful recruitment of 7-methylated guanosine nucleotide at the extreme 5' end of the eukaryotic mRNA takes place. Is it clear? So let me write it here. 7-methylated guanosine nucleotide, it successfully recruits at the extreme 5' end of the eukaryotic mRNA. And here what is 3' tailing here? In case of the 3' tailing, successful recruitment of multiple adenosine residues, they take place at the extreme 3' end of the eukaryotic mRNA. And here in order to recruit the multiple adenosine residues at the extreme 3' end of the eukaryotic mRNA, there is a requirement of a specific enzyme which is nothing but what? Poly-A polymerase, which is a template independent enzyme. Is it clear? So let me write it here. Multiple adenosine residues, they successfully recruit at the extreme 3' end of the eukaryotic mRNA by taking the advantage of poly-A polymerase, which is a template independent enzyme. So here after the successful organization of 5' capping and as well as 3' tailing, now here the eukaryotic mRNA, it is capable to export from the nucleus to the cytoplasm in order to participate in case of the translation. Now here the question arises, why the specific eukaryotic mRNA, it has to undergo 5' capping and as well as 3' tailing? Because in case of the cytoplasm, there is an availability of huge number of exonucleases. Is it clear? So let me write it here. Exonucleases. So here these exonucleases which are abundant in case of the cytoplasm, they are capable to cleave the extreme 5' end and as well as the extreme 3' end. So here in order to overcome the problem, in order to eradicate the problem, the specific eukaryotic mRNA, it has to undergo specific modifications like 5' capping and as well as the 3' tailing prior to the export from the nucleus to the cytoplasm. Is it clear? And the other specific strategy here it is nothing but what? Transport of mRNA from the nucleus to the cytoplasm. So here after the successful organization of RNA processing, the next specific strategy here it is nothing but what? Transport of the eukaryotic mRNA from the nucleus to the cytoplasm. So let me write it here. Transport of eukaryotic mRNA from the nucleus to the cytoplasm. From the nucleus to the cytoplasm. And the last strategy here is nothing but what? In order to participate in case of the translation mechanism. So here once when the eukaryotic mRNA, once when it is capable to export from the nucleus to the cytoplasm. Now here the specific eukaryotic mRNA, it has to participate in case of the translation mechanism. So let me write it here. Translation regulation. Is it clear? So here all these are the various strategies of the eukaryotic gene expression. Then after we will be studying regarding the concept of lac operon in detail. Is it clear? So let me erase the things here. So here in case of all the living organisms, the specific genes they get successfully expressed in order to perform one specific function. So here the gene expression it play an important role for regulating one specific function in case of all the living organisms. Is it clear? So let me write it here. In case of all the living organisms, specific genes get expressed in order to perform one specific function. So here in case of E. coli, if we consider in case of E. coli, right? E. coli. In case of E. coli, there is an availability of a specific gene which is nothing but what? LAC-Z gene. So let me write it here. LAC-Z gene. So here once when the LAC-Z gene, once when it undergoes expression during the transcription mechanism, it synthesizes a single stranded mRNA. And once when the specific mRNA, which generally synthesizes during the transcription mechanism, when it participates in case of the translation, it is capable to induce the expression of a specific enzyme, which is nothing but what? Beta galactosidase. Is it clear? So let me write it here. Beta galactosidase. It is the enzyme which generally synthesizes during the translation mechanism. Beta galactosidase. So here what is the role of beta galactosidase here? Beta galactosidase, it is the specific enzyme which is responsible for regulating the hydrolysis of lactose into glucose and galactose. Is it clear? So let me write it here. Lactose, which is a disaccharide, it undergoes hydrolysis in the presence of an enzyme known as beta galactosidase. So here beta galactosidase, it is the enzyme which is responsible for regulating the hydrolysis of lactose into glucose and galactose. Is it clear? Glucose and galactose. Now here the question arises in your mind. When the specific LAC-Z gene, when it undergoes expression here, in case of one particular E. coli organism, when the specific LAC-Z gene, it undergoes expression. Here the particular LAC-Z gene, it undergoes expression only if the particular E. coli cell, if it has lactose as the carbon energy source. Is it clear? If the particular E. coli cell or if the particular E. coli organism, if it has lactose as the carbon energy source, only under such condition, the LAC-Z gene, which is a part of the E. coli, it undergoes expression. And here in the absence of the particular lactose, the particular LAC-Z gene, it undergoes inhibition. Is it clear? So let me write it here. The specific LAC-Z gene undergoes expression only in the presence of lactose, and here in the absence of lactose, so let me write it here. in the absence of lactose, lag-Z gene expression undergoes inhibition, right? Inhibition. So here with this what we can conclude or what we can predict here is, for the successful regulation of the gene expression in case of the living organisms, here what are the various factors they play an important role here? The various factors which play an important role for the successful regulation of the gene expression in case of all the living organisms, they may include morphological features, physiological features and environmental conditions. Is it clear? So let me write it here. Gene regulation or expression in case of all the living organisms, is monitored and governed by morphological features, right? So let me write it here. Governed by morphological features, physiological features and environmental conditions. Right? And even in case of the particular living organisms, for the successful differentiation of the particular neonate stage to the adult stage, is it clear? For the successful differentiation of a particular neonate into the adult, here the specific genes, it plays an important role. Here the specific genes, they play an important role for the successful differentiation of a particular neonate into an adult organism. Is it clear? So let me write it here. So let me arrange the things here. For the successful differentiation of neonate into an adult organism, many of the genes, they play an important role. So let me write it here. For the differentiation of neonate into an adult organism, many of the genes, they undergo expression. Many of the genes undergo expression. And then after, we will be studying regarding how the gene regulation is monitored and what are the various components of operon in detail. Is it clear? So here let me arrange the things here. So here, in case of all the living organisms, control of the rate of the transcription mechanism, it is the predominant site for regulating the gene expression. So here transcription, it is the specific mechanism or it is the specific process through which the gene undergoes expression here. Is it clear? So let me write it here. In case of all the living organisms, control of the rate of transcription is the predominant site for regulating gene expression gene expression. Is it clear? So here the accessibility of the RNA polymerase enzyme in order to bind to the promoter region during the transcription mechanism, it is governed or it is regulated by the means of the positive regulator and as well as the negative regulator. Is it clear? During the transcription mechanism, the specific RNA polymerase enzyme, it is capable to bind to the promoter region by taking the advantage of specific regulatory elements like positive regulator and as well as negative regulator. Is it clear? So let me write it on the board. The accessibility of the RNA polymerase enzyme in order to bind the promoter region, is governed or it is regulated by the means of regulatory elements. And here the specific regulatory elements, they may include positive regulator and the other one is negative regulator. Is it clear? So let me write it here. Positive regulator and the other one is negative regulator. So here the positive regulator, here it is nothing but what? Activator. And whereas the negative regulator is nothing but what? Repressor. So here always the positive regulator known as activator, it has higher affinity to bind to the promoter region in case of the operon concept. Is it clear? So let me write it here. Positive regulator like activator has higher affinity to bind only to the promoter. Then in the same way, the negative regulator like repressor, has higher affinity to bind only to the operator. So here both the operator and as well as the promoter, they remain very adjacent to each other. Is it clear? Both the operator and as well as the promoter, they remain very adjacent to each other. And here each and every operon, it is having its own operator and as well as its own promoter. And always the particular positive regulator known as activator, it has higher affinity to bind only to the promoter, and whereas the negative regulator like repressor, it has higher affinity only to bind to the operator. Is it clear? So let me write it here. Both the promoter and operator remain very adjacent to each other. And here activator only binds to promoter, but repressor binds to operator. binds to operator. And then after we will be studying regarding the mechanism or the concept of lac operon in detail. So here what is lac operon and who were the two scientists who have explained the lac operon concept mechanism or the lac operon concept in detail. Is it clear? So let me arrange the things here. So here, if we consider the regulation of the lac operon, so here the particular lac operon for the very first time it was elucidated by two of the biologists like Francois Jacob and Jacques Monod. So here the lac operon concept, it was a collaborative work of two of the biologists like Francois Jacob and Jacques Monod. So here both of these biologists, they have explained or they have formulated the lac operon concept in order to explain the gene regulation or the gene expression in case of the various living organisms. Is it clear? So let me write it here. Lac operon was first elucidated by Francois Jacob and Jacques Monod. And here basically the particular lac operon, it contains a polycystonic structural gene unit. So let me write it here. Lac operon contains polycystonic structural gene unit, which is together termed as operon. So here the meaning of operon is nothing but what cluster of genes or group of genes. Is it clear? So here basically the particular lac operon, it contains three structural genes like LAC-Z, LAC-Y and LAC-A+. So the reason why here it is termed as what? Operon. So here basically the lac operon, it contains a polycystonic structural gene unit which is together termed as operon. So here likewise the lac operon concept, there are some more other additional operon concepts like tryptophan operon, arabinose operon and histidine operon. Is it clear? So let me write it here. Some other additional operons may include tryptophan operon, arabinose operon and histidine operon. Is it clear? And here if you look at the particular lac operon, so here basically the particular lac operon, it contains a specific gene known as what? Inhibitory gene. Is it clear? So here the I stands for what? Inhibitor, but not for the inducer. So here once when the particular inhibitory gene, once when it undergoes expression during the transcription mechanism, it synthesizes mRNA, and once when the particular mRNA, when it participates in case of the translation mechanism, it synthesizes a specific negative regulator known as repressor. Is it clear? And in addition to this, even the particular lac operon, it contains the specific structural genes like LAC-Z, LAC-Y and LAC-A+. Is it clear? So here once when the particular LAC-Z gene, once when it undergoes expression, it synthesizes a specific enzyme known as beta-galactosidase. So here what is the role of beta-galactosidase here? Beta-galactosidase, it is the particular enzyme which is responsible for the successful conversion of lactose into glucose and galactose. Is it clear? So let me write it here. Lactose successfully converts into glucose and galactose in the presence of an enzyme known as beta-galactosidase. Beta-galactosidase. Is it clear? Then in the same way, once when the particular LAC-Y, once when the particular gene, once when it undergoes expression, it synthesizes a specific enzyme known as what? Permease. So here what is the role of permease here? Permease is the enzyme which is responsible for the successful permeabilization of the particular sugar molecules into a particular biological cell. Is it clear? So here let us assume it as the biological cell. So here onto the interior side of the particular biological cell, for the successful permeabilization of the sugar molecules like lactose and as well as glucose, here the particular sugar molecules, they take the advantage of specific carrier proteins like permease. Is it clear? So here permease is the enzyme which is successfully regulated by taking the advantage of a specific gene known as what? LAC-Y. Then in the same way, once when the particular LAC-A, once when it undergoes expression, it synthesizes a particular enzyme known as what? Transacetylase. So here all these three are the three structural genes which are a part of the LAC operon concept. Is it clear? So let me write it here. LAC-Y gene, when it undergoes expression, it synthesizes permease, which is responsible for regulating the permeabilization of the sugar molecules into the biological cell. So here all the three structural genes which are a part of the LAC operon, they get successfully regulated in a coordinated pathway. Is it clear? All the three structural genes which are a part of the LAC operon, they get successfully regulated in a coordinated pathway. And here all these three structural genes, they play an important role in regulating the lactose metabolism. So let me write it here. All the three structural genes get regulated in coordinated pathway. And here these three structural genes are responsible for regulating lactose metabolism, So here for the successful regulation or for the successful expression of all the three structural genes which are a part of the LAC operon, here there is a requirement of a specific carbon energy source known as what? Lactose. Is it clear? So let me write it here. Lactose act as the carbon energy source for the expression of the three structural genes Is it clear? So here in the absence of the preferred carbon energy source known as glucose and in the presence of the particular carbon energy source known as lactose, all the three structural genes, they get successfully expressed in case of the LAC operon concept. Is it clear? In the absence of the preferred carbon energy source known as glucose and in the presence of the particular carbon energy source known as lactose, all the three structural genes, they get successfully expressed. That means to a particular living organism like E. coli, if the two carbon energy sources known as glucose and as well as lactose, if they get successfully exposed, always the particular E. coli, it shows higher affinity to intake only the particular glucose but not lactose, because glucose is monosaccharide but whereas lactose is disaccharide. So here the particular glucose which is a monosaccharide, it can be easily hydrolyzed or it can be easily degraded in comparison with the particular lactose which is a disaccharide. Is it clear? So here in the absence of the preferred carbon energy source known as glucose and in the presence of the particular carbon energy source known as lactose, all the three structural genes, they get successfully regulated or they get successfully expressed in a coordinated pathway in order to regulate the LAC operon concept. Is it clear? So here, always the particular inhibitory gene, once when it undergoes expression constitutively, is it clear? Always the particular inhibitory gene, once when it undergoes expression constitutively, are at all the times. So here the particular inhibitory gene, during the gene expression or during the transcription mechanism, it synthesizes mRNA. And here once when the particular mRNA, once when it participates in case of the translation, it is capable to regulate the production of a particular negative regulator known as what? Repressor. Is it clear? So here the particular repressor, as already mentioned, it has higher affinity to bind to a specific region in case of the operon concept, which is nothing but what? Operator. Is it clear? The particular repressor, always it has higher affinity to bind to the particular operator, which is a part of the lac operon here. So let me show it here. So here it is nothing but what? Repressor. Repressor, right? So here once when the particular repressor, once when it is capable to bind to the operator, or once when it successfully binds to the operator, by the time the RNA polymerase enzyme, it is unable to bind to the promoter. Is it clear? Once when the particular repressor, once when it is capable to bind to the operator, by the time the RNA polymerase enzyme, it is unable to bind to the promoter. So here once when the RNA polymerase enzyme, once when it is unable to bind to the promoter, here there is no chance for the expression of all these genes. Is it clear? So here the particular RNA polymerase enzyme, once when it successfully binds to the promoter, only under such condition, here the expression of all the three structural genes takes place in case of the lac operon. But here, once when the particular repressor, once when it successfully binds to the operator, here there is no chance for the binding of RNA polymerase enzyme to the promoter. So here in order to overcome the problem, here the particular lac operon concept, it takes the advantage of a specific inducer, which is nothing but what? Lactose. Is it clear? So let me show it here. Lactose. Lactose, right? So here the lactose is nothing but what? Inducer. So here once when the particular lactose, which is an inducer, once when it comes into the picture, so here the particular lactose, it is capable to hold up the particular repressor here. Is it clear? So here the particular lactose, which acts as inducer, it is capable to hold up the particular repressor. So here once when the particular repressor, once when it is bound by taking the advantage of lactose, which is acting as the inducer, now here the particular repressor, it is unable to bind to the operator. Is it clear? So here the particular lactose, which is acting as an inducer, it holds up the particular repressor. So here once when the particular lactose, which is an inducer, once when it holds up the particular repressor, by that time the repressor, it is unable to bind to the operator. So here once when the particular repressor, once when it is unable to bind to the operator, by that time the RNA polymerase enzyme, it is capable to bind to the promoter. RNA polymerase enzyme, it is capable to bind to the promoter. So here once when the RNA polymerase enzyme, once when it is capable to bind to the promoter, by that time the expression of all the three structural genes, it takes place. Is it clear? So here we need to understand that the particular lac operon, here all the three structural genes which are a part of the lac operon, they get successfully expressed only in the presence of lactose or allolactose. But here in the presence of specific sugar molecules like glucose and as well as galactose, so here all the three structural genes which are a part of the lac operon, they are unable to get successfully expressed here. Is it clear? So let me write it here. So let me arrange the things here. As already mentioned, so here the regulation of all the three structural genes, takes place only in the presence of lactose and allolactose. And here glucose and as well as galactose, they cannot act as the particular carbon energy source in order to regulate the expression of all these three structural genes. Is it clear? Here only the particular lactose and as well as allolactose, it acts as the carbon energy source for regulating the expression of all the three structural genes which are a part of the lac operon. And here glucose and as well as galactose, it cannot act as the carbon energy source for regulating the expression of all the three structural genes which are a part of the lac operon, because all the three structural genes which are a part of the lac operon, they are responsible for regulating the lactose metabolism. So the reason why here all the three structural genes which are a part of the lac operon, they are capable to work so efficiently or so effectively only in the presence of the particular carbon energy source known as lactose and as well as allolactose. But here in the presence of glucose, so let me write it here, glucose and galactose cannot act as preferred carbon energy source. Preferred carbon energy source. Is it clear? So here the regulation of the particular lac operon by taking the advantage of the repressor, here it is termed as what? Negative regulation. Is it clear? Here when the particular repressor, once when it successfully synthesizes from the inhibitory gene, here the particular repressor, it is capable to bind to the operator. So here once when the particular repressor, once when it is capable to bind to the operator, here there is no chance for the expression of any of the structural gene which is a part of the lac operon. So the reason why regulation of the lac operon by the means of the particular repressor, here it is termed as what? Negative regulation. So let me write it here. Regulation of lac operon by the means of repressor, here it is termed as negative regulation. Let me write it here, negative regulation. Negative regulation. And in addition to this, even the particular lac operon, it is capable to regulate in a positive manner, or it is capable to regulate in positive regulation. But here, right now, in your NCERT textbook, they have not mentioned regarding the positive regulation. And here you will be studying regarding the positive regulation of the lac operon in your higher classes. Is it clear? So here, this is regarding the concept of lac operon from the molecular basis of inheritance in detail. And in the next upcoming lecture session, we will be studying regarding genetic code in detail. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends.